Hello dear learners, myself Dr. Sanghamitra Chakraborty welcome you all to the video tutorial on acid, base and buffer under the aegis of OneMedicine. At the end of this session, you will be able to define the terms acid, base, buffer, pH and pK, enumerate the source of hydrogen ion in our system and describe how the pH is regulated by the buffers. Now, I know you all know what are acids and bases from your school age, but still, as per NMC competency, I need to say acids are proton donors. You can see here a prototype of acid HA, it dissociates to H plus and A minus, so this proton is donated. HCl, it dissociates to H plus and Cl minus, it is a strong acid, whereas carbonic acid, it is a weak acid. Bases are the proton acceptor, like ammonia, it reacts with hydrogen ion to form the ammonium ion. These are proton acceptors. What is pH? pH is the negative logarithm of hydrogen ion concentration. The arterial blood pH is 7.35 to 7.45. So, this is the biological reference interval of arterial blood pH. Till date, all the tests that have been discussed, they are done from venous blood, except this arterial blood gas analysis, it is done from arterial blood, and heparin is the anticoagulant of choice. The P stands for potence and pH, we will see it is a ratio, so it does not have any unit. Moreover, the pH ranges for any solution from 0 to 14, 7 being the middle point, less than 7 is the acidic pH and more than 7 is the alkaline pH. And this 0 to 14 range of pH, it is also known as the Sorensen scale. So, see here, an acid HA dissociates to H plus to A minus. And in terms of the equilibrium constant, this equation can be expressed as Ka equal to active mass of H into active mass of A minus anion divided by the active mass of HA. Taking negative logarithm on both sides, we have minus log Ka equal to minus log the product of active masses of H plus and A minus divided by active mass of HA. This reaction can be further simplified to pH equal to pKa log A by HA. This equation is known as Henderson-Hasselbalch equation. Now, if you have a look at this equation, and if there is a condition where the concentration of A equal to HA, then pH equal to PA, because this turns to be log 1 equal to 0, so pH equal to pK. So, what is pK? It is the pH of a solution where it is 50% dissociated or 50% undissociated, or at a 50% ionizable state. In other forms, pK is the negative base 10 logarithm of the acid dissociation constant of a solution. Now, what are the metabolic sources of acids which alter our blood pH? There are two categories of acids. One is the non-volatile or the fixed acid. Another one is the volatile acid. Non-volatile acids, they are mainly produced in various metabolic pathways like lactic acid, pyruvic acid, sulfuric acid, keto acid, phosphoric acid that are produced from the degradation of the phosphoproteins. And volatile acid, it is the carbonic acid which further dissociates to carbon dioxide and water by the action of the enzyme carbonic anhydrase. So, these are the sources of acids. Two types of acid, non-volatile and volatile, and non-volatile acids are also known as fixed acids. Now, what is buffer? Buffer is a solution which consists of a weak acid and its conjugate base that resists the change in pH. So, buffer, it is a solution of a weak acid and salt of its conjugate base which resists the change in pH even when a small amount of acid or base is added to it. Buffering capacity of a buffer depends on two important factors. One is concentration of the buffer, another one is pK of the buffer. The pK of the buffer close to the normal pH of our body, more the buffering activity will be of that particular solution. Maximum buffering capacity is shown at a range of pH which lies between pKa plus minus 1. This is the pH range where the maximum buffering capacity of any solution is shown. Buffers keep up H plus ion tied up until balance is re-established, so that the change in pH does not occur. So they keep the H plus ions tied up. Now, what are the types of buffer that are present in our system? They are present in the extracellular fluid, they are present in the intracellular fluid, and they are present in the RBC. Buffers that are present in the extracellular fluid are the bicarbonate buffer, phosphate buffer, or the albumin. To be more specific, better, in some book it is also given as protein as buffer. If you write jolly well, buffers in ICF or intracellular fluid, phosphate and bicarbonate, and buffers in RBC, that is hemoglobin. So these are the different types of buffer. We can see that both phosphate and bicarbonate, they are present both intracellularly and extracellularly, but their action differs in different compound. Bicarbonate is a more potent buffer extracellularly. Let us check. Most important extracellular buffer. So if we convert the Henderson-Hasselbalch equation, the equation turns to be like that, where H2CO3 equal to 0.03 into PCO2. PCO2 is the partial pressure of carbon dioxide in the arterial blood, and 0.03 is the diffusion coefficient. The ratio of bicarbonate to carbonic acid is 20:1 at optimal pH of 7.4, and it has a pKa at 6.3. There are two elements in the buffer system. If you look at the Henderson-Hasselbalch equation, there are two elements. One is the bicarbonate, another one is the carbonic acid. Bicarbonate is regulated by the renal system, and carbonic acid, it is regulated by the respiratory system or the respiratory physiology. In presence of strong acid, HCl, it will give the H plus, and this H plus will react with the bicarbonate to form the carbonic acid, which again will dissociate to carbon dioxide and water, and carbon dioxide will be exhaled by the lungs. In presence of strong alkali, this NaOH, it will react to form sodium bicarbonate. This bicarbonate is also known as the alkali reserve of the body. It is also known as the alkali reserve of our system. Phosphate buffer, it is the most important intracellular buffer. It has a major buffering role in the renal tubular fluid. The ratio of hydrogen phosphate to dihydrogen phosphate is 4:1 at optimal pH of 7.4. It has a pKa value 6.8. So it is more close to our physiological pH in comparison to bicarbonate. Still, bicarbonate is the strongest buffer of our system. It has two elements. One is the hydrogen phosphate, another one is the dihydrogen phosphate. Protein buffer, albumin acts as a buffer. Protein, what are protein? They are the polymers of amino acid that are linked by the peptide bonds. So protein contains both carboxylic acid and amino groups. Clear? This carboxylic acid it may exist as NH3 plus, or it may act as COO minus. So imidazole group of the histidine residue, which is present in the albumin, has a pK equal to 7.3. Each albumin contains 16 residues. Because of this histidine group, this albumin shows buffering capacity. Now, how does this buffer act? Let us take an example. So this is a structural or schematic representation of our lungs. This is the RBC within the tissues. So carbon dioxide that is liberated from the tissue metabolism that enters the RBC. It combines with the water and it is converted to carbonic acid. And this reaction is catalyzed by the enzyme carbonic anhydrase. Now this carbonic acid again dissociates to H plus and bicarbonate anion. The H plus, it combines with the oxygenated hemoglobin, and this oxygenated hemoglobin is reduced to form the deoxyhemoglobin. And this bicarbonate, it is taken out from the RBC, and in turn one chloride enters the RBC within the tissues. This is known as chloride shift. Now this bicarbonate, again it enters the lung capillaries. Within the lung capillaries in the RBC, the deoxygenated hemoglobin is converted to oxygenated hemoglobin, and the hydrogen that is liberated from the deoxygenated hemoglobin, that combines with the bicarbonate, carbonic acid is formed, and this carbonic acid, again by the action of carbonic anhydrase, it is converted to carbon dioxide and water, and carbon dioxide is exhaled out. Now this oxygenated hemoglobin, again it goes to the circulation and it enters the RBC. And in the lung capillaries, when the bicarbonate enters the RBC, again the chloride comes out from the RBC. So reverse chloride shift takes place. So this is how the bicarbonate buffer takes a role in controlling the pH system of our body. About 70 to 75% of the carbon dioxide that is released from the metabolism in the tissues, that is taken care of by the RBC without change in pH, and this is known as isohydric transport of the bicarbonate. So that's why bicarbonate is also known as the alkaline reserve of our body. So from this, it is very important as a short essay question in the university exam, the role of bicarbonate buffer. Try to prepare a note